Guys, welcome back to Social Academy. So in previous lecture, we were discussing about the translation initiation in the case of the prokaryotes, right? So today we are going to discuss in the case of the eukaryotic. So how in case of eukaryotes, the translation is getting initiation. So that is like, as we have discussed, there is something called PIC. So what is PIC? It is your pre-initiation complex which is present, okay? So this is in case of eukaryotes we're going to discuss. So we know in the case of eukaryotes we have got the 40S and 60S subunit. Okay, so let's begin with that. So first thing in case of eukaryotes is that eukaryotic ribosomes are recruited to the mRNA by scanning the 5' cap. Okay, so as in contrast to the prokaryotes, हमने वहां पे क्या देखा था, वहां पे कैसे शुरू होता है, they scan the RBS. Okay, so जब तक आपका RBS is not forming a base pair with your 16S rRNA, तब तक आपका initiation complex नहीं बनता है. Now in the case of the eukaryotes, it is the 5' cap which gets scanned. Okay. In eukaryotes, the small subunit is already associated with the initiated tRNA when it is recruited to the cap 5' end of the mRNA. So what does it mean? It means that to the small subunit, okay, आपका जो 40S subunit है, उसमें already आपका tRNA present है वहाँ पे, ठीक है? Now it then scans along the mRNA, of course in the 5'3' direction until it reaches your first codon. So थोड़ा सा difference है if you see in the case of the prokaryotes and the eukaryotic initiation of the translation. So this much is clear. So already in the smaller subunit you have got your initiated tRNA. Now what is going to be here the initiated tRNA? Normal आपका जो MET है, MET tRNA, sorry, tRNA MET, right? So MET means this tRNA is going to code, or it has got the anticodon for MET, methionine, and it is tagged or it is attached with the amino acid methionine only. Okay, जबकि हमने prokaryotes के केस में क्या देखा था, that additional 4-mile group was added. Okay, so in the case of the eukaryotes, it is normal methionine only. Now, what are the translation steps? So in case of eukaryotic cells, binding of the initiated tRNA to the small subunit always precedes association with the mRNA. So पहला step क्या है? That your small subunit of RNA, ठीक है, ribosome plus tRNA आपका सबसे पहले है. उसके बाद आपका mRNA आता है. Okay, so here you have got the sequence, whereas in the case of the prokaryotes, it was either पहले आपका tRNA भी आ सकता है और पहले आपका mRNA भी आ सकता है. But in the case of eukaryotic, it is first your tRNA gets associated with the smaller subunit of the ribosome, and then only your mRNA is coming. Okay, so you have got a separate set of the auxiliary factors which mediate the recognition of the mRNA. तो यहाँ पे आपको एक चीज ये समझनी है that when there is association of small subunit यानी कि 40S and tRNA, ये जब आपका प्रोसेस हो रहा होता है, साइड वाइज there is another recruitment of your mRNA. ठीक है, so mRNA में different proteins are there which goes in binds and help in the scanning of the stop codon. अब क्या होगा, later on these two together will come and forms your pre-initiation complex. Are you able to get this point? Yes, Tanvi, are you able to follow? यहाँ पे क्या हुआ है? So first it's your smallest ribosome along with the tRNA का complex बनता है. उसके बाद mRNA plus other proteins का complex बनता है. जब ये दोनों साथ में आते तब आपका pre-initiation complex बनता है. Is this much is clear to both of you?
Yes, ma'am.
Okay. Then the small ribosomal subunit which is bound to the initiated tRNA scans for the mRNA for the first AUG sequence. तब आपका ये जो PIC बना है यहाँ पे वो आपका first AUG sequence जो रहेगा start codon, it is going to scan. Okay. Now once it has been done, large subunit of the ribosome is recruited. Okay. After the initiated tRNA base pairs with the start codon. तो जैसे ही आपका start codon and जो tRNA का anticodon है base pair होगा, It means it is undergoing conformational change. Now, once that conformational change happens, there are certain initiation factors which will get removed and your larger subunit will form. जैसे कि हमने लास्ट टाइम देखा था IF3 जो था इनिशिएशन फैक्टर 3 इन द केस ऑफ द यूकैरियोट्स, ये क्या करता था? इट वाज प्रिवेंटिंग द बाइंडिंग ऑफ लार्जर सब यूनिट, राइट? तो उसी तरह से यूकैरियोट्स में भी वी हैव गॉट सर्टेन इनिशिएशन फैक्टर व्हिच प्रिवेंट्स द बाइंडिंग ऑफ द लार्जर सब यूनिट. जैसे ही आपका फर्स्ट बेस पेयरिंग होता है बिटवीन द कोडॉन एंड द एंटीकोडॉन, स्टार्ट कोडॉन एंड द एंटीकोडॉन ऑफ द tRNA, it undergoes conformational change which results in the removal of those initiation factors and then आपका जो large subunit है वो आता है. ठीक है? I hope this much is clear. So this is just the beginning, like to understand like how in the case of eukaryotes the initiation is happening. Okay, तो ये था story. अब क्या करेंगे हम इस चीज को detail में पढ़ेंगे ना. When I say detail, it means hell lot of proteins are there which we are going to study now. Okay, so let's go slowly. I don't want you guys to get confused. So before I proceed, so as I mentioned in the previous lecture also, that whenever you have got small e written, it means it is for your eukaryotes. ठीक है, confuse नहीं होना है because IF1, IF2, IF3 आपका prokaryotes में भी present है. So there it is just your IF1, initiation factor 1, initiation factor 2 and 3, so on. In order just to differentiate between the eukaryotic and prokaryotic proteins, we have got small letter E, which is for your eukaryotes. ठीक है, इतना clear है. Okay. Now EIF1, A, 3 and 5. हमने prokaryotes में क्या पढ़ा था? 1, 2 and 3, right? यहाँ पे थोड़ा सा difference आएगा. So look at the figure also. So what do we see over here? That to your smaller subunit जो आपका 40S है, ठीक है? You also have that EPA का sections, correct? Now to this EPA 40 subunit, you need to see that how and when which proteins are binding. Okay, so together EIF1, 1A and 5, okay, they are going to act in analogous to IF3 and IF1. अगर आप ध्यान दो, IF1 का क्या काम था? To prevent the binding of charged tRNA at the amino acyl site. ठीक है, so that directly your first initiated tRNA comes and sits over here. तो 1 का काम था to prevent this and 3 का काम था to prevent binding of the large subunit, if you remember. ठीक है, अब यहाँ पे क्या हो रहा है? इनका काम करने के लिए the function of IF1 and 3 is being done by EIF1, EIF1A and EIF5. ठीक है, okay. If you see, can you see that over here you have got the 1 as well as 1A and 5. Can you see the locations clearly? So you can see that prevention of tRNA is being done by 1A. Is it clear? Prevention of binding of large subunit is done by 1 and 5. Okay. Now the initiated tRNA is escorted to the small subunit by 3 subunits GTP binding protein EIF2. This is similar to your prokaryotic IF2 which is bound to GTP. ठीक है, तो क्या करेगा? वो यहाँ पे आके बाइंड होता है, राइट? इफ यू रिमेम्बर, इस केस में क्या होगा? यहाँ पे this EIF2 GTP is bound to your initiated tRNA. And this entire thing is brought and placed onto the P site. Okay? Now this entire thing is known as your ternary complex. Guys, are you able to follow? इतना याद हुआ आपको?
Can you explain the last step again, ma'am?
Yes. So initially we have spoken about the function of EIF1, A and 5, which is analogous to your prokaryotes का IF1 and 3. ठीक है? Okay. Now we have got another protein which is eukaryotic IF2 bound to GTP. If you remember, in the case of prokaryotes, we had IF2 bound to GTP. So, जिसका hydrolysis was making sure that IF1 is released, okay, and it becomes GDP then. So when it is GDP, it loses its affinity, okay, and then IF2 is also released. Similarly, in the case of the eukaryotes, you have got EIF2 GTP, which goes and binds to tRNA. This is your initiator tRNA, correct? You can see I is written over here. So it means it's your initiator tRNA charged with amino acid methionine, okay? Now this is being recruited with the help of EIF2 GTP. Now you can see it over here. So this 43S pre-initiation complex means what? It has got your initiation factors 1, 1A, 5, 3, 2. Can you see in the figure? So this entire thing is known as your PIC or pre-initiation complex. Why it is called 43S? Because 40 तो आपका smaller subunit का हो गया, rest 3 इनका भी weight है. Okay, so this becomes your 43S PIC, pre-initiation complex. Is it clear this much?
Yes, ma'am.
Okay.
Yes.
Now, okay. So for case of the eukaryotes, the initiator tRNA is charged with the methionine and not the formal methionine. So this we have already discussed. Now, EIF2 position Z met tRNA I met in the P site. So इसका क्या काम है? To bring that initiator tRNA at your P site, peptide site, and total it forms your PIC. Okay. So it is noteworthy that EIF3 is almost as large as tRNA. एंटायर 40 है, तो इसका पूरा टोटल वेट हमने 43 दिया हुआ है. ओके, थोड़ा सा यहाँ पे कट हो रहा है बिकॉज ऑफ द फिगर. ओके, नाउ सो एज आई सेड, जब वहां पे आपका स्मॉल सब यूनिट इनिशिएटेड टीआरएनए वाज गेटिंग एसोसिएटेड, on the other hand your mRNA is getting associated with other initiation factors. ठीक है? Okay. So in a separate series of reaction, the mRNA is getting prepared in order to recognize your small subunit. So पहला क्या होगा? So the 5' cap is being recognized by your EIF4E. ठीक है? ये आपका क्या हो गया? Initiation factor 4E. Okay. So then EIF4G, it is going to bind to both E and your mRNA. क्या करता है? It goes and recognizes your 5' cap of the mRNA. ठीक है? G क्या करता है? It is going to recognize your E as well as mRNA on G. Okay? We'll go slowly, don't worry. Okay? Now, अब A क्या करता है? EIF4A. So it is going to bind your G and mRNA. So G क्या कर रहा है? E and mRNA. A क्या कर रहा है? G and mRNA. Okay. So all these associations, okay, it is important so that the overall the translation can be controlled. So the association of EIF4G and E is important because the overall level of translation in the cell is controlled at this step. अगर हम later, like once we complete this topic, you will see that the regulation of your translation will be controlled at this stage, EIF4G and A. ठीक है? Okay. Now, so entire this complex. So this complex is joined by 4B, which activates the RNA helicase activity of EIF4A. So A क्या है आपका? Helicase है. ठीक है? So it is going to unwind any secondary structures which is present in the mRNA so that it gets ready for your translation. Now let's go slowly again because मुझे पता है इतनी आसानी से ये याद नहीं होगा. So what we have discussed first, यहाँ पे देखो, see at the second part what you need to remember is in this case, okay, 1 second. Yeah. So in this case, when your mRNA is getting prepared, you just need to remember that it is your eukaryotic initiation factor 4. ठीक है? इतना याद रहेगा? Now let's see what ABCD is coming. So first thing is that your E comes and recognizes the cap of the mRNA. Clear? Then G, which comes and recognizes the E and mRNA. Okay? A, पहले E आया, फिर G आया, फिर A आया. A क्या कर रहा है? It is recognizing your G and mRNA. ठीक है? Now, what B is doing? So the entire complex of E, G, A and B is formed along with the mRNA. This complex is joined with the help of the B. So B क्या करता है? It is going to activate the activity of A. A का क्या activity है? RNA helicase activity. Okay. So this will get activated only if you have got any hairpin loop structures or any secondary structures present on the mRNA. अब वो हमें क्यों चाहिए? Because if those secondary structures are present, then it becomes difficult to scan your RNA, okay, for your translation. अगर आप ध्यान दो figure में, what do you see over here? That your start codon is present over here. That is after this hairpin structure, the secondary structure, correct? अगर ये structure नहीं हटाएंगे, so it will become difficult for your ribosomes, difficult for your tRNA to base pair with the start codon. Yes, are you able to follow, guys, this much? So this is happening in parallel to what your smaller subunit and tRNA association. So ये एक दूसरी जगह पे हो रहा है and sideways आपका smaller subunit and tRNA is also getting associated. Is this much clear?
Yes, ma'am.
थोड़ा सा tricky है because there are so many initiation factors coming into play, right? So finally, all these interactions are there among all these initiation factors, and it forms your total. So now, what is this है? See, पहले क्या था आपका 43S pre-initiation complex बना था, correct? Now, if you see these two together, when this part, what we have discussed now, the smaller subunit along with the tRNA and other initiation factors, plus mRNA and another initiation factors together, they become 48 pre-initiation complex. So this is your 48 pre-initiation complex. ठीक है? Guys, clear?
Yes, ma'am.
हाँ? I mean, I hope you're getting the idea कि क्या हो रहा है, because obviously it will take more time and revision for you guys to remember which initiation factor is doing what and binding where. ठीक है? Apart from that, I hope the concept is clear कि कैसे proceed हो रहा है आपका initiation of translation. Correct? I hope that's correct.
Yes, ma'am.
Okay, so together at the end, it is forming your 48S pre-initiation complex. Okay. Now, translation initiation, it is going to, in translation initiation factors, they hold the eukaryotic mRNA in circles. इसका क्या मतलब हुआ? So once you see that, like in order to facilitate the good translation or the efficiency, the enhanced efficiency of the translation, these initiation factors are actually helping, okay? And because of these interactions between the initiation factors, they are keeping the mRNA in the circle. Okay, कैसे हो रहा है? Let's see that. So the presence of poly A tail contributes to the efficiency of the translation. In addition to binding the 5' end of the eukaryotic mRNA, अभी हमने क्या देखा? That your eukaryotic initiation factor 4E is coming and binding to the 5' end of the mRNA, correct? Similarly, we have got proteins which is known as poly A binding protein. Okay, किसी textbooks में ये directly PABP लिखे होते हैं. That is nothing but your poly A binding proteins. So ये क्या होगा? mRNA में at the end we know that around 200 nucleotides of A is present, right? So this poly A binding proteins will go and bind to this poly A tail of the mRNA. Okay. Look at the figure. So यहाँ पे हमने अभी क्या discuss किया था? That you have all this 43 pre-initiation complex, then mRNA आएगा bind करेगा. So mRNA के bind करने के बाद आपको 48 का पूरा मिलता है. ठीक है? Now what do we see over here? That along with the poly A tail, can you see over here? Poly A tail is there. ये आपका पूरा mRNA का 5' है, ये आपका 3' है, right? So what do we see? यहाँ पे आपका initiation factors है, and यहाँ पे आपका poly A binding proteins है. And these poly A binding proteins are again interacting with the initiation factors present over here, right? Now, because of these interactions, like these interactions, what is present over here, you can see that the mRNAs are present in a circle. ठीक है? अब आपसे क्वेश्चन ये आपके सामने आता है, like what will be the significance of this? क्यों? Instead of keeping straight like this, जहाँ पे आपका 5' है, 3' है, यहाँ पे आपके सारे initiation factors हैं, and यहाँ पे आपका poly A tail है, राइट। इसको सर्कल में रखने का क्या फायदा हुआ? If you ask me, any guesses? Tanvi, Akshaya, would you like to try? Like, what is the benefit of keeping the mRNA into a circle? What do you think? Try करो, क्या हो सकता है रीजन? Yes, Tanvi, would you like to try? Akshaya, you? No one? Okay, no problem. See, the thing is that when you keep the mRNA into a circle, what happens is that the reinitiation of translation becomes easy. So as I said before also, the same mRNA can be translated again and again, जहाँ पे you can get several copies of your protein, right? Now for this to happen, if you could keep the tail, जहाँ पे जैसे ही यहाँ पे termination होगा, okay, the stop codon will be present. The moment it is going to stop, if this is kept near, what can happen then? Immediately the ribosomes will come and bind over here and they are going to start the translation. Okay, so for the reinitiation of the translation, if you keep the mRNA into the circle, it becomes enhanced overexpression of the proteins, or it will make the translation efficiency good. Okay, so I hope by this you will get the point. Okay, now the start codon is found by scanning the downstream from the 5' end of the mRNA. अभी क्या है? We have learned till now कि 5' mRNA end cap has been scanned. ठीक है, poly A tail भी आपको मिल गया है जहाँ पे poly A binding proteins are associated, right? Now the second thing which is important is that downstream of the 5' cap there is your start codon. अब हमें उसको scan करना है, ठीक है? Now how this is being done, let's discuss that. Okay, so once assembled at the 5' end of the mRNA, the small subunit and associated factors, they are going to move along the mRNA. Okay, in an ATP dependent process. So what does it mean? It means it is your ATP requiring step, energy requiring step. Okay, now this is being done by 4'B associated RNA helicase activity. Just now we have discussed about it, right? So during this movement, the subunit, the small subunit is going to scan the mRNA for your first start codon, and that is your AUG. Okay, so scanning of the start codon is energy dependent, and this energy is provided by the EIFOB. अगर आपको याद है अभी हमने डिस्कस किया था, ठीक है? So the start codon is recognized through the base pairing between the anticodon and the initiated tRNA का start codon. अब क्या होगा? The moment this scanning is done and there is a base pairing between the anticodon and the start codon. So what will happen? This will result in the conformational change. The moment anticodon and the codon of their start codon base pairs, okay, all those three nucleotides are forming base pairing. So there is going to be a conformational change. Now this conformational change will result in the release of your initiation factors 1 and 5. अब क्या होगा? This will also result in the this conformational change. Okay, this conformational change will result in the hydrolysis of the EIF2 bound GTP. Now what will happen once the GTP hydrolysis happens? This is going to lose its affinity for the EIF2. Now what will happen? All these initiation factors will no longer will have affinity for the smaller subunit. All this will be getting released. ठीक है, तो अभी तक हमने क्या पढ़ा? दैट 5' कैप जो है आपका mRNA का स्कैन हुआ, ठीक है? All these initiation factors were brought together. Now once the tRNA forms the correct base pairing with the first start codon, what will happen? There is a conformational change. This conformational change will result in the removal of or reduce affinity of these initiation factors like EIF1 and 5 and 2. ना अब क्या होगा, अब ये सारे सब यूनिट्स हट जाएंगे, ये सारे सब इनिशिएशन फैक्टर्स हट जाएंगे. सो व्हाट विल हैपन नेक्स्ट, देन once all these are removed, then your what will happen is that this entire thing is going to recruit for your larger subunit. ठीक है. सो वंस यू हैव गॉट द रिलीज, सी आपने स्कैन किया यहाँ पे, ठीक है, ऑल दीज आर एटीपी डिपेंडेंट प्रोसेस. नाउ ऑल द स्कैनिंग यू फाउंड दैट करेक्ट बेस पेयरिंग इज हैपनिंग. कैन यू सी इन द फिगर here, the correct base pairing is happening between the tRNA and the start codon. Now once this has been done, it will release in the removal of your 1 and 5 and EIF2, okay, which was bound to GTP. अब ये सब हटने के बाद क्या होगा, you will have another initiation factors coming, that is 5B GTP. Now this is going to bring your larger cell. हाँ. Yes.
Ma'am, your voice is breaking.
At what point, Tanvi, in this slide? Over here, from where it started breaking?
When you started this next slide, this one slide.
Okay, okay. So I'll just repeat now. Am I clear now? My voice is clear now?
Yes, ma'am.
Okay, so I'll just repeat this slide again. Okay. Okay. So what I was saying was that once we are starting the scanning of the start codon. Okay. So when the scanning is being done, okay, now so what is happening during the scanning? What we are waiting for is that आपका tRNA और स्टार्ट कोडॉन के बीच में बेस पेयरिंग होना चाहिए. ठीक है. एक बार जब ये बेस पेयरिंग हो जाता है, this results in the conformational change. This conformational change will bring about release of all these factors. Okay. And later what will happen is that the another GTP bound initiator protein is there, that is your EIF5B, which is bound to GTP. ठीक है. Now, इसका क्या काम है? इसका काम है to bring your larger subunit. Now, which one is your larger subunit over here? The 60S subunit. Okay. So, what will happen? Since before EIF1, 5, ये सब आपके यहाँ पे present थे, if you see, so it was blocking the association of the larger subunit with the smaller subunit. Now, जैसे ही आपका यहाँ पे GTP hydrolysis हुआ, conformational change हुआ, EIF1, all these were released, correct? So now there is nothing which can prevent the association of the larger subunit. So this is being brought by EIF to 5B. So once this comes, okay, what will happen? The entire complex now it becomes ATS, जो हमें मालूम है, correct? Now your ribosome is ready. आपके पास उसके larger subunit है. Okay, I'll just go to the next slide. The figure is there, so you can see it over here. Okay, so जहाँ पे ये release हुआ आपका 60S subunit आया, it forms the complete association. Now once this subunit is attached over here, it will also remove the rest of the initiation factors. Now you have got the entire subunit. Okay, here this is ready. You have got the ribosomes, you have got the initiator tRNA, base pair with the start codon, correct? So everything is placed accordingly which is required. आपका ये first में है, and you have got the removal of these initiation factors, right? अब क्या है, आपके पास empty A site भी है, empty A site. What does it mean? Now it is available for your new amino acylated charge tRNA. ठीक है. So depending on the codons which is present over here, you are going to have the recruitment of the new tRNA, जो भी आपका amino acid यहाँ पे होगा. Again, if you see in the figure, okay, if you see in the figure, you will see that there is a kink present again. As we have mentioned before, this is required so that the codon can be read in 3 nucleotides. ठीक है. This much is clear, guys. Are you able to follow?
Yes.
Yes, ma'am.
Okay, good. Of course, you need more practice. Like आपको ये sequence याद रखना पड़ेगा, like which initiation factor is coming and binding into what. क्यों important है? Because they are going to ask at which step, if suppose there is an inhibitor which is binding and blocking the activity of EIF5, ठीक है? तब आपको समझ में आना चाहिए, if you block the release of this particular initiation factor, what step are you blocking in the translation? So what will be the answer? So if you block the release of this, so the new charge tRNA cannot come, and hence the translation is getting prevented. ठीक है? तो इस तरह के क्वेश्चन आएंगे. If you go back, अगर आपसे ये पूछा जाए, if there is a inhibitor which is blocking the release of your EIF2, so what is the answer? Of course, the initiation of the translation is getting prevented, but you need to know at which step what is happening. Okay, so if I say that this is there, they might ask you, so they might ask you like whether the additional or whether the large subunit was added or not added in the initiation complex. आपको मालूम होना चाहिए अगर IF2 ब्लॉक है तो ये भी आपके ब्लॉक रहेंगे, तो जिसकी वजह से क्या होगा, आपका लार्ज सबयूनिट ऐड नहीं हुआ है. ठीक है, so that's why all these the sequence by which these initiation factors are coming and performing their functions, it is important to know. Okay, now so at the end you have got your entire ATS complex which is going to be performing the translation. Okay, now just going back, so here I have listed out all the similarities between the prokaryotes initiation factors and the eukaryotes initiation of the translation. Okay, so हमने कैसे देखा, IF1 prokaryotes and eukaryotic IF1A, they bind to the A side. ठीक है, तो इसका क्या काम है, to prevent the charge tRNA coming prematurely. Okay, the function of IF2 is split between the two initiation factors in the case of the eukaryote. So IF2 का कौन कौन काम कर रहा है, F2 हो गया, 5B हो गया. इसका क्या काम था, it mediates initiated tRNA recruitment and detects the pairing with the initiator, the start codon. Okay, now 5B क्या करता है, it is helping in the recruitment of the large subunit. Similarly, they regulate IF3 हो गया यहाँ पे. IF3 क्या काम करता था, which was preventing the binding of the large subunit and all, right? If you remember, so ये काम के लिए आपने हमारे पास यहाँ पे ये बात. ठीक है, so if you can just go through, एक बार याद हो जाएगा, या figure if you keep side by side, so you can remember like how these functions are being done. Okay, yeah. So let's do certain questions now. So here the initiation of translation of eukaryotes is also over. ठीक है, थोड़ा सा आइडिया मिला आपको कि कैसे क्या है. Like just watch the videos again later and you can understand like the differences between the prokaryotic and the eukaryotic initiation of the translation. ठीक है, so I believe everything is clear till now. So far so good, guys. Akshay, Tanvi. Yes, कोई डाउट है आज के सेशन में? ओके, ठीक है, आई एम श्योर आपको याद करने को टाइम लगेगा, बट दैट्स ओके. सो डिपेंडिंग ऑन दोस विल हैव द क्वेश्चंस लेटर. ओके, okay, so there are few questions, like I think 2 or 3 questions are there based on the previous topic, so let's discuss those. Okay, so it's based on the transfer RNA and amino acid tRNA. Directly I have taken from the PDF which I have shared with you guys. Okay, so let's do this question. So your first question here, it says, consider the following statements. First statement, tRNAs are charged by the attachment of an amino acid to the 3' adenosine nucleotide via high energy acyl linkage. Okay. पहले देख लेते क्या पूछा है. Which of the following statements are correct? So let's go like that only. So guys, what do you think? It is true? तो ये क्या कह रहा है, आपके पास tRNA है, 3' end में आपका amino acid charge होता है. Is it correct? With via high energy acyl linkage. Okay, what do you think, guys? It's a correct statement? क्या लगता है? Let's see. Okay, एक बार सारे पढ़ लेते हैं हम लोग. So amino acyl tRNA synthetases charged tRNA in 2 steps. Okay, adenylation in which amino acid reacts with GTP, and step 2 is tRNA charging in which adenylated amino acid reacts with tRNA. Okay. Third statement, most organisms have 35 different tRNA. Each amino acid tRNA synthetases attaches a single amino acid to only one tRNA. It attaches single amino acid to only one tRNA. tRNA synthetases recognize acceptor arm and anticodon loop of cognate tRNAs. Same amino cell tRNAs uses same नहीं है, sorry, some है. It use an editing pocket besides catalytic pocket to charge tRNA with high accuracy. अब इसमें क्या पूछा है? What are the correct statements? Okay, yes, all of you, both of you, Akshay, Tanvi, you have to respond. Yes, क्या होगा इसका आंसर? You can go in the other way around also, like जो नहीं है statement, like the statements which are wrong, you can, based on that also you can select the answer. So which statement is wrong? Have I ever mentioned that the organisms have 35 tRNAs and हर एक tRNA के लिए हर एक amino acid है और vice versa? Have I ever mentioned like that?
No, ma'am.
Okay, so from that statement it should be clear that C आपका गलत statement है. Okay, so option आपका 1 भी गया और 2 भी गया. What about A, D, E, A, B, E? तो इससे भी आपको मालूम हो जाना चाहिए, यहाँ पे दोनों है, सो A करेक्ट है और E करेक्ट है. ओके, अब आपको बस ये डिसाइड करना है B और D में से कौन सा सही है. ओके, टेल मी. अक्षय, क्या होगा इसका आंसर? इट्स वेरी सिंपल गाइस.
I feel B is correct.
Okay,
Option.
okay, Akshay is saying B is correct. Okay, ये अक्षय क्या है? What about you? तन्वी, तुम्हें क्या लगता है?
Yes, ma'am. Maybe B.
Okay, both of you are wrong. क्यों? If you remember, there was a figure in which I have shown that apart from the catalytic side, there is an editing pocket in the amino cell tRNA which helps in the high accuracy. So this is because of this editing pocket. ठीक है? It is the amino cell tRNA synthetases which make sure that the tRNAs are charged with the correct amino acid. क्योंकि आपको याद हो तो मैंने बताया है ribosomes cannot differentiate between the wrong amino acid tRNA. Correct? So ये एक reason है. एक और चीज, अगर आपको ये भी नहीं मालूम है, सपोज दिस स्टेटमेंट यू डोंट नो, लाइक अबाउट द एडिटिंग पॉकेट ऑफ द tRNA synthetases, I agree that it adds, okay, it is going to charge the tRNA in 2 steps and first step adenylation है. ठीक है, आपको ये ध्यान में देना चाहिए, जब मैं एडिनाइलेशन बोल रही हूँ तो इसका मतलब क्या है? इट इज द एडेनिन ग्रुप. करेक्ट? एडेनिन ग्रुप आपको कहां से मिलेगा? यू आर गोइंग टू गेट इट फ्रॉम एटीपी. बट यहाँ पे उसने क्या बोला? एडिनाइलेशन इन विच द एमिनो एसिड रिएक्ट्स विद जीटीपी. जीटीपी से क्या मुझे एडेनिन मिल सकता है? नहीं ना. फ्रॉम जीटीपी आई विल गेट गुआनिन ग्रुप. तो सेंटेंस में ही आपका था आंसर कि एटीपी आई कैन नॉट. एडेनिन, एडिनाइलेशन मींस अटैचमेंट ऑफ दैट एडिनो जो हमने पढ़ा था, राइट? तो वो आपका एमिनो एसिड में कैसे अटैच होगा? इफ द जीटीपी इज प्रेजेंट. जीटीपी से गुआनिन मिलता है, गुआनिलेशन हो सकता है. तो यहाँ पे आपको समझ जाना चाहिए था दैट इट इज रॉन्ग स्टेटमेंट. समझ में आया मिस्टेक?
Yes, ma'am.
Yes, ma'am.
ओके, गुड. सो कैसे हमने सॉल्व किया? सारा पढ़ने की जरूरत नहीं है, जस्ट सी विच वन यू थिंक वेरी श्योर दैट विच इज रॉन्ग स्टेटमेंट. उसके बाद आप सिर्फ ये देखो 4 की जगह 2 ऑप्शन हमें देखने हैं. ठीक है? सो यू कैन डू इन दैट वे ऑल्सो, दैट मेक्स इट मोर इजी. आपका टाइम सेव होता है उसमें. Otherwise, these big questions, they are going to take so much of time. Okay, again, it's all about amino cell tRNA synthetase reaction. Consider the following statements. So these many statements are given. Now you need to find which all statements are correct about the amino cell tRNA synthetase. Okay, again. So what do you think? What about A and B? कौन सा सही है इसमें से?
A.
Correct. बी आपका गलत है. लुक एट द ऑप्शंस नाउ. सो वी सी दैट बी यहाँ पे दिया हुआ है. सो ये स्टेटमेंट आपका नहीं है. ओके, देन नेक्स्ट. तो हमने क्या देखा? अभी ए है करेक्ट, ओके, बी है गलत. अभी आगे जाने की जरूरत ही नहीं है. व्हाट डू यू थिंक व्हाट विल बी द आंसर ओवर हियर?
Fourth.
Fourth one.
हो गया, आपको पूरा पढ़ने की जरूरत ही नहीं थी. बट स्टिल अगर आपको लगता है कि हाँ मुझे थोड़ा सा पढ़ लेना चाहिए, लाइक टू बी श्योर अबाउट इट, देन यू कैन जस्ट रीड अबाउट इट. कौन सा था? ए क्या है आपका? ई क्या? ए ई एफ. ए ई एफ. सो इट्स अ मिक्स एनहाइड्रेट बॉन्ड इज फॉर्म. हाँ बनता है. असाइल एसिड बॉन्ड इज फॉर्म. हाँ बनता है, राइट, बिकॉज एट द एंड हमने बोला ना इट्स अ हाई एनर्जी असाइल लिंकेज इज फॉर्म. सो दैट इज आल्सो प्रेजेंट. करेक्ट है. बीच में आपका एनहाइड्रेट बॉन्ड फॉर्म होता है जब आपका एडिनाइलेशन होता है ना. if suppose I want to, if you want to see, अगर आपके पास बहुत टाइम है, ठीक है, if you think you have time and you just want to check, तो आप क्या कर सकते हो? सी और डी पढ़ो. सी में क्या दिया है? एमिनो एसिड इज अटैच्ड टू दी 2-3- हाइड्रोक्सिल ऑफ द न्यूक्लियोटाइड फैक्टर 82. क्या ये सही है? नहीं. टीआरएनए सिंथेसिस क्या करता है? 2-3- किसका ऐड करता है? ऑफ दी एडिनोसिन जो आपका एंड में प्रेजेंट है, राइट. बट कोफैक्टर को नहीं करता है. इट इज दी 3' आर्म में जो प्रेजेंट आपका ACC वहां करता है. आर यू एबल टू फॉलो? थोड़ा डाउट आया क्या? कोई कंफ्यूजन है? इफ यू रिमेम्बर, एमिनो एसिड कहाँ अटैच होता है आपका? टीआरएनए में जो आपका 5'CCA है, करेक्ट. इसके 2-OH या 3-OH में आपका एमिनो एसिड अटैच होता है. एग्री?
Yes.
Yes, ma'am.
यहाँ पे क्या दिया है? इट्स अ कोफैक्टर. यहाँ पे क्या है? कहाँ अटैच होता है? एक्सेप्टर आर्म ऑफ योर टीआरएनए, करेक्ट. इट इज नॉट अ कोफैक्टर. कोफैक्टर कब यूज होता है? जब आप एमिनो एसिड्स को एडिनाइलेट कर रहे हो. सो दिस स्टेटमेंट इज आल्सो रॉन्ग. ना एमिनो ग्रुप ऑफ द एमिनो इज एक्टिवेटेड, कुछ ऐसे अलग नहीं होता है वहां पे. ओके, देन व्हाट ऑल वी हैव गॉट? थायोसिन बॉन्ड नहीं है, ये बॉन्ड नहीं है. सो दिस इज हाउ विदाउट रीडिंग द एंटायर ऑप्शन आल्सो यू कैन गेट द आंसर. तो हमने क्या किया? जैसे हमने पढ़ा ओके जीटीपी से कोफैक्टर नहीं, ये रॉन्ग है हमारा. तो जैसे ही मैंने देखा कि हाँ इट इज प्रेजेंट ओवर हियर एंड ओनली ऑप्शन वेयर ए इज आल्सो प्रेजेंट इज द करेक्ट आंसर. तो हमें आंसर यहाँ मिल गया. ओके. So these are the tricks I'm telling you. Without reading the entire question, you can get the answer. Is this question is clear?
Yes.
Yes?
Yes.
Okay. Now there was another question. ये तो बहुत ही simple लगा मुझे. Like it was funny also. So the question says, suppose you mix the following components of the protein synthesis in a test tube. Okay. Amino acids from rabbit, ribosomes from dog, tRNA from mouse, mRNA from chimpanzee, and all the other necessary enzymes from the giraffe. If protein synthesis happens, which animal proteins will be made? किसका होगा? Rabbit, mouse, dog, chimpanzee. किसका क्या आंसर होगा इसका?
Chimpanzee.
Correct, chimpanzee. क्यों? Because इसका mRNA is going to code for your protein. Correct? बाकी आप एमिनो एसिड कहाँ से लाए, टीआरएनए कहाँ से लाए, इट डज नॉट मैटर. It is the information which is present in the mRNA which is going to get converted to the proteins. और ये कहाँ से लिया है? mRNA किसका लिया है? Of the chimpanzee. So you get answer 4. Correct. Yeah, that's all. So I hope you are getting the idea कि किस तरह के क्वेश्चन पूछे जा सकते हैं, right guys?
Yes, ma'am.
Yeah, so we'll be doing this type of exercise every lectures. ठीक है. So after each जो भी हम डिस्कस करते हैं उसके बाद we are gonna do all these exercises. ठीक है. जैसे यहाँ पे जो क्वेश्चन आ रहा है we are going to get it. If you think that आपको मालूम है ये questions या if you want like these questions should be discussed in the lecture, so you can just WhatsApp me कि मैम ये वाले questions हम लोग डिस्कस करते हैं. Otherwise मुझे क्या करना पड़ेगा? फिर मुझे सारे लेने पड़ेंगे. And these questions, जैसे अभी जो चिंपांजी वाला क्वेश्चन था बहुत सिंपल था, राइट? उसको नहीं भी लेती तो चल जाता है. So if you guys get time and all, go through the PDF and let me know what all questions needed to be taken seriously. ठीक है? Okay then. चलो then I'll be stopping here now.